Okay, so I might begin just by giving a little bit of a recap of what we've gone through this morning. So our first presentation by Janine gave us some fantastic insight in defining network nodes. And then we've had two presentations, once we've defined nodes, in actually quantifying connectivity between pairs of nodes. So Rob just gave us an overview of mapping connectivity, structural connectivity using diffusion MRI. And prior to Rob, we had Shella Kelsholz, who gave us an overview of mapping functional connectivity and some of the challenges of mapping functional connectivity. So in the second part of the education course, we're now going to look at or consider the case once we've actually mapped connectomes. What are the kind of analyses that we can do, and what are some of the steps that we should consider before we go ahead and run graph analyses or compare connectomes between groups. So in the next half an hour or so, I'm going to consider three key issues that can be performed or that can improve graph analyses. And I'm first going to focus on thresholding connectomes. I'm then going to go through the often overlooked issue of null models. And finally, I'll give you an overview of some methods for comparing connectomes between groups. My name's Andrew Zalesky. I'm from the University of Melbourne, and as I've mentioned already, I'm co-organising the course with Alex Fornito. So to begin with thresholding, what is thresholding? In a nutshell, thresholding is really about eliminating the weakest connections in a connectome, and these are the connections that are most likely to be false positives. There are various reasons to threshold, but I really want to emphasise that thresholding is not an essential step. We can proceed with analysing a weighted connectome without performing any thresholding. But some of the reasons to threshold are that thresholding can improve the clarity of connectome visualisation, it can reduce false positive connections, and it can also simplify the interpretation of some graph analyses. For dense connectomes that are mapped at the resolution of individual vertices or voxels, thresholding becomes essential to reduce the storage burden of such connectomes. And finally, there are some studies that have shown that thresholding connectomes can improve the strength of associations with behavioural measures. But of course, on the flip side, there are many reasons not to threshold a connectome. First and foremost, the thresholds that we use are typically arbitrary, right? We don't have a ground truth understanding of what the connection density is of a brain network. Is it 1%? Is it 10%? Is it 20%? We don't know. So the threshold that we choose is typically arbitrary. There's also some recent work by Sevier and colleagues that's shown that actually removing the weakest connections using a thresholding approach doesn't really have much of an impact on connectome measures. As I've shown here on the right-hand side, I've shown a graph measure called path length, and we can see in the unthresholded network the path length has a value of 2.34. If we then go ahead and threshold the network, the value of path length doesn't change very much. So the argument here is why do we want to even bother adding this complexity into our connectome mapping pipeline if it doesn't have a large overall impact on the pipeline itself. Once we've thresholded, or if we've decided to threshold our network, another step that we can do is binarise the network. And when we talk about binarisation, we're only interested in the presence or the absence of a connection. And our connectivity matrix only comprises ones and zeros. So in the bottom matrix there on the right, the white cells correspond to ones and the black cells are zeros, the absence of any connectivity. And the advantage of binarisation is that we know that connectivity strength or variation in connectivity strength can potentially be due to confounds and the biases that Rob and that Shella have already pointed out. And so by binarising the connectome, we can overcome to a certain extent those biases. But on the flip side, this variation in connectivity strength is important, right? We know that connections vary in strength by orders of magnitude, and we want to preserve those effects in our network. And by binarising that, we are effectively suppressing that information. So we're losing that rich variation in connectivity strength across the network. So these are some careful choices that need to be considered. Should you threshold your connectome? And if you decide to threshold, should you also binarize your network before conducting graph analyses? And there's no right or wrong answer here. This largely depends on the kind of analysis and the context of the analysis. So here's a conceptual example of thresholding. This is a toy network. We're starting off with a connection density of 26%. So connection density is, we use that to quantify the severity of thresholding. It's just the actual number of connections in our network normalized by the total possible number of connections. And in this network, we have 3 types of connection strengths. There's weak, moderate, and strong. So in the first step, we can remove the moderate connections, and that gets us to a connection density of 20%. Then we can go one step further and remove not only the moderate connections, but also the weak connections, and we end up with a connection density of 13%, as shown on the bottom. And what you'll notice at the bottom, one of the nodes becomes fragmented from the network, right? That orange node at the bottom is disconnected. And this is a problem for the computation of several graph metrics, and I'll come back to this issue in a moment. You're probably also wondering that it's biologically implausible that we have certain brain regions or neurons that are completely disconnected from a nervous system. So this is one of the issues that can come up if we threshold too severely. So now let's get to the methods of thresholding. Broadly speaking, there are 2 kinds of thresholding methods that have been proposed in connectomics. One is density thresholding and the other is weight-based thresholding. And these are really simple. With density thresholding, we specify a desired connection density, in this case 20%, and then we incrementally remove the weakest connections in the network until we achieve our desired connection density. The other method is called weight-based or weight thresholding. And in this case, rather than specifying a connection density, we specify a minimum connection weight, whether that be streamlines or a correlation coefficient. So in this example, we say we only maintain connections that comprise 3 or more streamlines. Now, with these 2 methods, an interesting property is that they are essentially the same in the sense that if you specify a connection density, you can always find a corresponding weight that will give you or that will yield that connection density. And the converse holds. If you specify a weight, you can always find a corresponding connection density. And so you might ask, why bother then? Why are you considering 2 separate methods if these are effectively one and the same? Well, the difference between these 2 comes about when we start to look at groups of individuals. And in this case, I'm going to give an example where I've only got two individuals, Alice and Jack, to illustrate the difference between weight and density thresholding. So I want you to note for Jack's network, it comprises a greater number of stronger connections. These are the thicker edges in the network compared to Alice. So if we were to go ahead with weight-based thresholding and we impose a threshold of eliminating moderate and weak connections, we see that if we look at the thresholded networks on the right, Jack's network comprises a greater number of connections. In other words, the connection density is higher in Jack's network compared to Alice. Now, the problem with this, if we were then to go on and do some kind of graph analysis, say, for example, comparing path lengths or clustering coefficient, we might find differences between Alice and Jack that are, in a sense, trivially due to the difference in connection density. So it might not be a good idea to compare networks that differ substantially in connection density. And so this is a disadvantage of weight-based thresholding. So you say, okay, let's go to density-based thresholding, right? If we use density-based thresholding, by definition, the thresholded network will necessarily comprise the same number of connections between Jack and Alice, and therefore we overcome that confound, right? We can't say, well, if we find a difference between Jack and Alice, that's just because one of the networks comprises more connections, which is a confound of our thresholding approach. But the disadvantage of density-based thresholding is that we've needed to lower the bar for Alice's network, right? We've had to admit some of these moderate connections into the network, and these can potentially be spurious or false positive connections because they're weaker. So this is an important consideration in group-based studies. Should we use density or weight-based thresholding? And to give you an example of that, I'll consider the case of schizophrenia. We know when we're investigating functional connectivity in schizophrenia, connectivity is generally reduced in individuals with schizophrenia. So with weight-based thresholding, as I've shown in the example here, we end up with a density of 53% for the patient with schizophrenia, whereas the healthy control, we have a density of 75%. So this is a weight-based threshold of 0.2. Any correlation below 0.2 is eliminated from the network. We only look at those that are above 0.2. And so this would present a problem for a between group comparison. In contrast, if we go to density thresholding, we overcome that issue, right? We end up with the same number of connections in the patient and in the healthy control. However, as you see there on the bottom, I've had to lower the weight threshold for the patient. I've had to lower it to 0.31, a correlation of 0.31, just to make up those numbers, to ensure that we've got a matched number of connections between the patient and the control. So there's no right or wrong answer, and there is some more advanced thresholding techniques and approaches that have been proposed to deal with this problem. Check out the reference there at the bottom for more details. Another critical point is that the choice of threshold. As I've said, we don't really know. We don't have a ground truth understanding of what the true connection density is of a nervous system. So I always recommend it's important to consider or to repeat the particular analysis across a range of thresholds. In this case, I've shown a connection density on the x-axis and then our specific network measure on the y-axis. Once we've repeated the analysis for a range of thresholds, we can then consider a summary measure such as the area under the curve or even just the average across the different connection densities. And that takes us away, at least to a certain extent, from the dependence of the threshold choice. This also relates to a method called topological data analysis. Have a look at the work by Sizemore and colleagues for details. I won't have time to present that here. There's also an approach called multi-threshold permutation correction, which in a similar way enables us to consider a range of connection densities and casts the problem in a statistical framework. I won't have time to go into the details here, but check out the paper by Drake-Smith for further details. I also wanted to touch on the method of consensus thresholding, which is an important thresholding method, and this is where, rather than thresholding networks individually, we rather look at a consensus across individuals. And here we have two thresholds. We have a weight threshold and a group threshold. And the basic idea is that we want to keep connections that are consistently found across a group of individuals. So if you see there, for Alice's network, one of the connections that is weak, one of the very thin connections, is maintained in the network. And that's because if we look at Jack's network and if we look at Bob's network, that particular connection is strong. And that's why we maintain that weak connection. Of course, one of the disadvantages of this is that consistency in connections across a group of individuals doesn't necessarily mean that connection is a true positive, right? We can have systematic errors that occur across individuals. And this approach has also been extended to take into account distance dependence effects. If we want to preserve long-range connections or other connections with certain properties, the consensus thresholding approach can be adapted, and I've given some references there if you want to check out those methods in more detail. Thresholding can also be cast as in a statistical framework, and if this is specifically in the case of functional connectivity, if we're measuring functional connectivity using the correlation coefficient, these correlation coefficients can be converted to z-scores, and we can then have a p-value for each connection, and so we can threshold based on a p-value. Say any connection that's less than 0.05 is maintained in the network, and other connections are discarded. One thing to be cautious of here, though, is that these p-values are often too optimistic because of something called autocorrelation in the Bolt signal. And so there are various approaches to overcome that issue of autocorrelation and derive p-values that are more realistic. So check out the work there, the references are at the bottom for further details. What I've considered here are only global thresholding methods. This is where we have one threshold and we apply that threshold to all of the connections in an individual's network. There is, however, other thresholding methods referred to as local methods, and this is where we consider different thresholds within certain locales of the network, and this can be used to emphasize certain topological structures, although I should say these methods haven't been widely used in the field of connectomics. And some examples of these methods are the disparity filter, as well as multi-resolution thresholding methods. And finally, I'd want to introduce the minimum spanning tree. If you remember before, I showed you that if we threshold severely, certain nodes can become disconnected from the network, and this is not biologically realistic. The minimum spanning tree is one way to avoid nodes becoming disconnected nodes. And the basic approach here is that we first find the minimum spanning tree, which is it's a tree. It ensures that all nodes are connected by definition, and then we incrementally add connections to the minimum spanning tree until we achieve our desired connection density. So to summarize, connectome thresholding involves eliminating the weak and potentially spurious connections from our connectomes. Thresholding is not essential. We can proceed doing all of our graph analyses without any kind of thresholding and by analysing weighted networks. However, it can help in alleviating false positives, ease computational burden and storage requirements. It can simplify the interpretation of some graph measures, and it can also improve the clarity of connectome visualization. I've given you an overview of weight and density thresholding and given you some examples of how they can differ in a between-group comparison and some of the things that might need to be considered in between-group comparisons of thresholded connectomes. And I also want to reiterate the importance of evaluating a range of thresholds, particularly given that we don't know the ground truth connection density of a connectome. And there's some further reading there at the bottom. And by the way, I forgot to mention that you'll see in my slides I've given reference to functions from the Brain Connectivity Toolbox. If you want to implement any of these thresholding methods, you've got the relevant functions there. There's also a Python version of the BCT with equivalent functionality as well for those using Python. So I now want to turn to the topic of null models, and this is often overlooked in the field of connectomics, I think, as well as neuroimaging more generally, but it is a really important topic because it enables us to determine whether certain effects that we're seeing in a connectome can be separated from noise or from chance level occurrence. I want to motivate the importance of null models using a simple example, using the measure of path length. And path length, as probably most of you know, path length just tells us how many hops or how many connections do we need to traverse to get between two nodes. So in this example, the two yellow nodes, the path length is 2, and I denote the path length with L. So we have a connectome, we compute the path length, we find out that it's 2, and so we ask ourselves, well, is a path length of 2, is that short? Is it typical, or is it particularly long? How do we know that? How do we understand what this path length value is telling us. And a null network or a reference network can address that question. So what we typically do is we consider, well, what's the path length in a random network that comprises the same number of nodes and the same number of edges? And for random networks, we have an analytic expression for the average path length, and it's the expression that I've shown there. So we can plug in the number of nodes, which is 9 in this case, and we can plug in the number of edges in our network, which is 15, and the formula tells us that in a random network, the average path length is 2. And so we can say for our toy network there on the left, the path length of 2 is typical. It's not particularly long or it's not particularly short. And so in this case, the random network is serving as our null model or our reference model. What about a lattice? What about if we did the same thing in the lattice? We can take a lattice here with 20 nodes and 40 connections, and we see that the average path length is 2.5. Is it long? Is it short? Or is it typical for a network to have a network of this size to have a path length of 2.5? So we can do the same thing, right? We take our formula, plug in 40 for the number of nodes, 20 for the number of nodes and 40 for the number of connections, and we get a path length of 2 once again. And so we can say that the path length in our lattice network is quite long, right? So lattices are not particularly conducive to efficient communication or to shortest paths. So this gives us an example of our null network is matching the number of nodes and the number of connections in our observed network, in our actual connectome. And you might be saying, well, why do we only match the number of nodes and connections? Why don't we also match, for example, the node degree, or for example, connection lengths, network geometry, other higher order properties? What should our null model preserve? What aspects of our null of the real network should be preserved in the null? And it's actually it's not an easy question to address. And here's a bit of a conceptual overview of how null models could be understood. So the outermost concentric circle here comprises the set of all random networks, right? And this is probably not a very intuitive or helpful null to consider. And so we can then add in further and further properties into our null network. So for example, we can only consider random networks that are matched in the number of nodes. That's the blue circle. Or we can say matched in the number of nodes and edges. Or we can say matched in the number of nodes, edges, and the node degree. So node degree is just the number of connections that come out of a node. And this is actually what we use most commonly in connectomics. It's the same number of nodes, the same number of edges, and the same node degree. And so we say, does our observed network differ in that case? And this can be generated using an approach called Maslov-Snepen rewiring. More recently, there's been some interest in also preserving geometry of the null. So this enables us to test whether geometry has an effect on the topology of our networks. So if we keep adding more and more properties to our null network, we end up converging to our actual network. So how do we use null networks in practice? How would we actually run a test to see whether our network shows a property more so than expected by chance? And what we typically do here is determine the null space. So in this example, it's the salmon-coloured space where we're preserving nodes, number of nodes, edges, and node degree. And we'd randomly sample networks from this space. We'd compute our measure of interest, which in this case is path length, and then we compute the average path length. And if we take the ratio of the path length in our real connectome, in our empirically measured connectome, to the average of our sample networks, this gives us a ratio. And so if that ratio is greater than 1, we can see that path lengths in our observed network are particularly long. If it's equal to 1, path lengths are the same as our random network, and if it's less than 1, we can see that path lengths in our actual network are quite long. Sorry, I said that the opposite way around, right? It's that if the ratio is greater than, if the ratio is less than 1, it means that path lengths in our real network are shorter than what we'd expect by chance. We can also do this in a statistical sense, and we can compute p-values to test whether a certain property is observed more so than in our benchmark. I won't go through the details of that here. So as I said, in connectomics, we typically preserve the number of nodes, edges, and node degree, and we can do that using a very simple approach called Maslov-Snepen rewiring. Basically, we choose a four-node motif in our network, and we just rewire it, as I've shown there. Rather than the two connections going from UV to IJ and IJ, we rewire it such that we have UI and VJ, and we repeat this rewiring step again and again and again, hundreds of times until we achieve sufficient randomization. And if you think about it for a while, this will guarantee that the node degree is preserved and the number of connections remains the same, right? In this rewiring step, we're not losing any connections. However, this approach can't be applied to weighted networks, or there are heuristics that can be used, but they don't necessarily preserve strength. But check out the work by Rubinov and Sporns if you're interested in the weighted versions of this. So here's an example of Maslov-Snepen rewiring in practice. Here I'm considering the macaque connectome and a mouse connectome. The one on the left here is the empirically measured connectome. I've then shown a sample of a null network. This was generated using Maslov-Snepen rewiring. And on the right there, I've shown the ratio of three measures: the clustering, the path length, and small worldness. And so we see there for both the macaque and the mouse that the clustering has a ratio that's greater than 1. So we can see that these networks are more clustered than what we would expect in our random network. The path length is roughly 1. So path length is comparable to what we see in the random, in the null network. And these are the characteristics that would diagnose small worldness. And so we see there also that the small worldness coefficient is greater than 1. So both these networks are small world networks. So I would also like to mention that more recently there's been a lot of work on preserving the geometry in our null networks. And this geometry, by geometry here I mean the length of connections. This could either be the length of an axonal, of a connection estimated using tractography, or the Euclidean distance between nodes. And I won't go into the mechanics of the approach, but what we see here is if we preserve, or if we generate random networks, null networks that preserve geometry, it becomes harder to beat the null. So the blue line there, this is if you look at the bottom right here, I'm showing the clustering coefficient as a function of connection density for a particular human connectome. And we see that the blue line at the top that's labelled connectome, that's the value of the clustering coefficient in the real connectome. I've also shown the case for a non-geometry preserving null, which is the red line. So this is nothing but the Maslov-Snepen rewiring algorithm. And the green line shows the case of a geometry preserving null, right? And so you can see that the geometry preserving null gets closer to what we see in for the empirical connectome. So this tells us that, at least to a certain extent, the geometry of the connectome is explaining this topological property of clustering. Briefly, generative models they're not really null networks per se, but they enable us to generate connectomes in silico. I'm not going to say too much about this, but they're based on a very simple wiring rule where we can, a probabilistic wiring rule where we have a distance constraint as well as a topological rule. And by combining these two in a multiplicative way, this gives us a probability of adding a connection between a pair of nodes. And then we can incrementally add connections according to this rule and generate networks that recapitulate key properties of empirically measured connectomes. And the topological rule that yields the best match with empirical networks is the matching index, which is effectively saying that we want to add connections between pairs of nodes that share the same neighbours. So just to recap, null networks set expectations for network organisation under certain null hypotheses. In connectomics, the most widely used null network is where we preserve the number of nodes, the number of edges, and the node degree. And we can generate null networks that preserve those three properties using the Maslov-Snepen rewiring algorithm. So this is basically, we take our real network, we apply Maslov-Snepen rewiring, and this generates multiple samples, multiple random networks that preserve the three properties. More recently, there's been interest in preserving the geometry in our nulls, and generative models can be used to build null networks in silico. So in the last part of this presentation, I'm going to give a very brief overview on comparing connectomes and some of the statistical methods that can be used to compare connectomes between groups. So the basic idea is that we've mapped connectomes for a group of patients or a group that have been subjected to an intervention. We also have a comparison group, and these connectomes could be functional connectomes or structural connectomes using the methods that Rob has already touched on. And there are two kinds of approaches that we can do here. One is connectome-based inference. And this is where we essentially want to identify particular features of the connectome that differ between the two groups, or that are associated with a behavior, or that are modulated by treatment. And another, or a more recent approach is based on prediction, connectome-based prediction. And this is where we look at the connectome as a feature space, and we want to train a model to predict, for example, treatment response, or to predict some kind of clinical outcome. So I want to briefly consider both inference as well as prediction. And the challenge here is that connectomes are high dimensional, right? Even if we're using a connectome with 100 or so nodes, as Scheller has already pointed out, we're dealing with potentially thousands of distinct connections. And so how do we deal with this? How do we perform inference and build these into a predictive model? This just recaps that point. We can actually perform prediction at a global level. This is where the connectome is characterized by a single global metric, such as efficiency, modularity, the Ridge-Club coefficient, as we'll learn later on this afternoon about these particular graph measures. The problem with the global comparison is it doesn't give us any specificity, right? We just have one value to characterize the whole connectome. So it doesn't tell us about the specific region or the particular node that might show a difference in a particular patient group, or that associates with a certain behavior. So this is really easy to compare statistically, but it doesn't give us good specificity. We can also do comparison at the level of nodes. This can be done using node strength, efficiency, or, for example, node-based clustering. And at the top of the hierarchy here is connection level comparison. And this is where we consider each connection individually. And this could be using any of the measures that Rob has already pointed out, or if we're dealing with functional connectivity, the measures that Scheller pointed out this morning. So I'm going to focus specifically on connection level inference, and that's because this is the most challenging level of inference, and this is sometimes referred to as connectome-wide, and a CWAS, or a connectome-wide association study. So in this approach, we have a hypothesis, and we test that hypothesis at each connection comprising our network, and then we're left with a massive multiple comparisons problem, right? So we've got a p-value and a test statistic at each edge, and this could potentially be hundreds or thousands of different connections. So there are various generic methods for correcting for multiple comparisons. I'm sure many of you are already aware of these. The Bonferroni correction, which is probably too stringent here. There's also the false discovery rate, which is a little more lenient. However, we know that in connectomes that the effects that we find are generally clustered to form networks. So here I've shown two scenarios. The scenario on the left, where the effect of interest, so this could be, for example, connections that are weaker in strength in schizophrenia, or some connections that are associated with some kind of behavior. In scenario one, these effects are scattered throughout the network, whereas in scenario two, these form a connected cluster, or a subnetwork. And this is more likely, the effects are more likely to be distributed as shown in scenario two, and we see that across multiple disorders there, as I've shown in the panel on the right. Now, the point that I want to get across here is methods such as the false discovery rate or Bonferroni correction don't care about that. They offer no more power in scenario 2 than scenario 1. They don't distinguish between these two scenarios. But we can use methods. There are methods that are developed that can give us greater statistical power for scenarios where effects form clusters or form subnetworks. One of those approaches is the NBS, or the network-based statistic. To give you a bit of a rundown of how that works, effectively we set a threshold or a minimum meaning effect size, and we throw away all the connections that have a test statistic or an effect size that is below our desired threshold. And in the remaining set of connections, we try to identify subnetworks. So subnetworks are just clusters of connections. And in this example, we have a subnetwork that comprises 5 connections. And then we ask ourselves, what's the chance of actually finding a subnetwork comprising 5 connections by chance? And we can assess this probability using permutation testing. So effectively, if it's a between-group comparison, we can shuffle patients and controls and recompute this procedure. And as I've shown there on the right, I've shown the maximum subnetwork size across these different permutations. And so you can see here, finding a subnetwork of size 5 is exceedingly rare. And so based on this null distribution, we can actually compute a p-value for finding a subnetwork of size 5, and in this way perform statistical inference. There are also other methods for performing inference in CWAS designs. I'm not going to go through these in detail, but check out the publications. Here at the bottom, there's matrix factorization, spatial pairwise clustering, CCA, as well as numerous other inference methods. And this is indeed an active area of research. So in the remaining 5 minutes, I now want to turn to connectome-based prediction. This is a little bit different from inference. This is where, rather than identifying features that differ between a group, we consider the connectome as a feature space, and we want to train a model to predict some kind of outcome. This might be treatment response, diagnostic status. And so this is effectively a cross-validation where we take the upper triangle, put this into some kind of a machine learning model, prediction model. And on the right here, I've shown the performance of various models. And in this work, it was found that using non-sparse regularization, so in other words, L2 regularization, yielded the highest prediction accuracy. So the more we go to the right of the plot, the higher the accuracy. There are also methods such as connectome-based predictive modeling, which are connectome-specific approaches for performing prediction. So some tips if you're training a connectome-based prediction model. One thing to consider is this concept of a tangent space, which can improve prediction accuracy. So rather than using partial correlation or correlation, if you map your connectivity to a tangent space, this can improve prediction accuracy, although the improvement is modest. It doesn't make a huge difference. It's also important to remember that error bars on accuracy estimates can be really large, and that's particularly the case for small sample sizes. So this, once again, points to the importance of sample size in machine learning models in connectomics. Some other key points is use k-fold cross-validation if possible, rather than leave one out, cross-validation. Evaluate the area under the curve. Be cautious of data leakage. Make sure that your connectomes in your training group don't end up somehow being placed within your test group, because it will inflate your prediction accuracies. And it's also important to evaluate, if possible, to evaluate accuracy across multiple sites and scanners, and this will ensure that your model is generalizable. It's not specific to a particular type of scanner or a particular kind of potential confound at a given site. Here's some software if you're interested in running connectome inference and analysis on connectomes. There's a whole range of software that is available for you to use. Most of the functions that I showed earlier on in my presentation on thresholding and null models are available in the BCT, the Brain connectivity toolbox, and the BCT is available both as a set of MATLAB functions as well as Python functions as well. So to wrap up, some resources. If you want some more details, check out our book. This is with Alex Fornito and Ed Bullmore, Fundamentals of Brain Network Analysis, where we go through these issues in much more detail. That's all from me. So happy to take any questions. No questions?
Hi, thanks, Andrew. Did you ever, when I do my analysis, I sometimes encounter like an interaction between the thresholding selection procedure that I use and the randomization. Can you comment on this a little bit?
Sorry, I missed the second part.
Oh, sorry, like an interaction between the thresholding and the randomization procedure that I choose.
Regularization in what?
The randomization.
Oh, randomization.
So when I use Maslov-Sneppen, for example, then you can have a network configuration which is actually not easy to randomize.
Right. Yeah, look, I mean, I think in a way, these are separate issues, right, in terms of the randomization and the thresholding. Do you mean like an interaction between the actual threshold choice and the randomization? So if the thresholding choice is particularly stringent, so if you have a very sparse network, there's not really a lot to randomize, right?
Exactly.
Or if we have differences in the density across a group of individuals, it might be possible to randomize a certain individual to a greater extent. These are the
exactly. Now, if you take the MST, for example, as the ultimate maximum type of thresholding that you can use.
Yeah.
In Maslov-Sneppen, it is not randomizable.
Yeah.
Word, but you can't randomize it. So,
I agree.
Also, when we use the scripts for the BCT, it will produce a random network back, but it will just be the same network.
Yeah. I mean, I think it's an important issue also to evaluate the extent of I mean, is your network really randomized, right? We run Maslov-Sneppen as a black box, but when we have our randomized network, how do we really know if it is randomized to enough for our purpose? I mean, it's ultimately an iterative procedure, and then we need to stop at some point. And yeah, I haven't given much thought to the issue of interactions with our threshold, and I don't know if there's any work out there that specifically looked at that interaction between the threshold choice and the actual randomization. Yeah, the randomization method or the extent of randomization. Something for future research, perhaps.
We'll come back to that next year.
Yeah.
First, thank you very much for your excellent presentation. My question is a little bit off topic, and it's about null models. There is a lot of literature of null models, but there is also an uprising of Bayesian methods for inference in other contexts. So not using only a null model, but also an alternative model. So you compare which one is more probable. So it's more informative. Do you know of any of these methods which apply to networks or to neuroimaging in general? Does it make sense also?
I think absolutely. I mean, null models is one way to do this, but certainly using model comparison is, I wouldn't even say alternative, right? It's a complementary approach that can be used. And I think this reminds me of work, say, for example, in ERGMs, exponential random graph models, where the approach taken is exactly as what you've pointed out, right? Model comparison and determining which model provides the greatest likelihood. So absolutely, I think that they're complementary, and there's certainly scope here. I mean, I haven't covered that here, but it's certainly an alternative way to address this issue.
Great, thank you very much.
Hi, I might have missed this sort of point, but the tools that you talked about are applicable to both structural and functional connectomics. The reason I'm asking for that is usually when I do some sort of thresholding on structural, it's easier to sort of get rid of the weak connections and your connectome metric sort of ends up with a bunch of zeros and so. But when you do it on functional connectivity, then you still end up with some sort of values. But it is either the activation or deactivation, and then you go ahead and sort of try to interpret that. So there are some, I guess, differences between those. I was just wondering what you think about that.
Yeah, it's true. There are subtle differences between functional connectivity and structural connectivity, and I haven't gone into those subtleties here. Speaking very broadly, is that the functions that are available in BCT can be equally applied to functional connectivity and structural connectivity, but you're exactly right. When applying these methods, it is important to consider the characteristics of the actual measure of connectivity that you're using. So, for example, streamline counts. If we're looking at raw streamline counts, these are discrete values, whereas if we're using functional connectivity with the correlation coefficient, these are continuous and they range between -1 and 1. So these particularities of the actual connectivity measure need to be taken into account when thresholding. So yeah, you're absolutely right.
Sure. Thank you.
Yep.
Okay, thank you very much for your brilliant talk. I wanted to ask you about task connectivity. So in essence, when you have an event-related task fMRI, you usually see that task CPPI is advised, for example, in the contour box. Mm-hmm. And I'm thinking what would change here, that you have events and it's a more complex, let's say, design. Mm-hmm. Would you try to correct in a similar way? And also I see Turoi hypothesis-driven models. Usually you just have a fewer selection of nodes and a more simple multiple comparison. What would be your recommendations?
Yeah, look, I mean, task-based connectivity is a whole different area. I haven't considered that at all, but there are particular methods, I think, that, as you pointed out, things such as PPI, to understand task-based connectivity and separate task-specific effects from the effects of rest or effects that are not of interest in the experiment. And also, yeah, if you have specific hypotheses about particular regions of connections, of course, that they should be used to constrain the search space rather than looking across the whole brain. We can certainly constrain the search space by looking at particular nodes or particular connections. Yeah, but in general, the methods here would, if we've got a task-based measure of connectivity such as PPI, and we can map a network, a whole brain network, the methods that we have here would still be applicable to that measure of connectivity. Of course, as with the other question, there are specifics with task-based connectivity which would need to be taken into account. But broadly speaking, these we can threshold and we can perform statistical inference on task-based networks.
Thank you.
Perhaps the last question, so I don't want to hold up people for lunch.
Hi, thank you so much. I'm also interested in task-based connectivity, but one thing I've also been interested in is how these networks we define behave over time. And you've gone over some really, you've given a really clear presentation of different ways that we could define networks, but could you comment a little bit on different ways that we could look at how these networks shift and change in form over time?
Yeah, I mean, the idea of time-varying connectivity is really important. There's a lot of work that has been done in this area over the last couple of years, and one way to model this, at least from a graph perspective, is multi-layer networks, right? Where we have, rather than just having one network representation, we can have multiple networks where the network connectivity and even the nodes themselves can change as a function of time. Of course, that introduces a lot of complexity into the analysis, right? We've got to then determine the weights that we use to interconnect these different layers of the network, and analysing a single connectome can be challenging. So once we've got multiple layers, that introduces the storage burden and the complexity of the analysis. But I agree, it's a crucial area, understanding the dynamics of connectivity and how to best represent those dynamics in terms of a model, whether that be a multi-layer network or some other kind of representation. So I think we might leave it there and break for lunch. So we've got a 45-minute lunch, and perhaps we come back at 12:45 for the afternoon session.